Hello students, welcome to Capman Academy, and today we are going to talk about indirect method of gene delivery, which is also known by the name vector or virus mediated gene transfer, or known as biological method biological method or known as Agrobacterium mediated gene transfer. Right, so for this method, Agrobacterium tumefaciens is used, which is a gram negative bacteria, rod shaped as you can see in the image, and causes crown gall disease in the plant. Right, so it survives as a pathogen in the presence of a suitable host. Now we are talking about the Agrobacterium. It has different type of biovars. It exists in different strains. So the most common biovars of the Agrobacterium are: number 1, Agrobacterium tumefaciens. This one is called biovar 1. Second is the Agrobacterium rhizogens. This one is called biovar 2. And third one is Agrobacterium vitis. And this one is called biovar 3. Now they have the specific type of specificity. Due to that, they were actually used for the gene transfer. Agrobacterium tumefaciens contain plasmid, and rhizogens, they contain plasmid. Right, and vitis, they are same like Agrobacterium tumefaciens and they contain plasmid. And as we all know, plasmids are the extra chromosomal DNA part and they were used for the gene cloning purposes. Right, so now we are going to talk about the TI plasmid. So as the name suggests, TI plasmid is your tumor inducing plasmid. Right, and TI plasmid, it consists of two regions. Number 1 is your tDNA and second one is your Vir region, as you can see here. Right, so TI plasmid is actually have tDNA region and Vir region. So the tDNA region, it is used for the transfer into the host. It has left border and right border. So this whole region is actually transferred into the host plants. Another region, which is your Vir region, this one, it helps in tDNA transfer. It helps in tDNA transfer in target host. So now we are going to discuss about these two regions. And now let's talk about the first region, which is your tDNA region. And tDNA region, as you can see in this image, it is flanked by two border sequences which are called direct repeats, and they are consist of 25 base pairs and known by the name left border and right border. Right? And within these borders, they contain some specific genes for number 1, auxin, second for cytokinin, and third for opine. Right? Auxin, cytokinin, and opine. So auxin here, it is used for the production of uncontrolled cell division, right? Which leads to the tumor in the host, and then this tumor leads to the disease which is called Crohn gall disease. Cytokinin here, it also helps in the uncontrolled cell division with the auxin, and this one also participates in the tumor formation. Opine, if we talk about, it acts as or provides the nutrients. It provides nutrients to the bacterial growth. Right? Second region is your Vir region. So Vir region, it contains different type of virulence genes. Virulence genes. And it has nearly 11 operons and can code for 35 proteins which are called Vir proteins. Vir proteins. Right? And out of 35 Vir proteins, 20 are involved in the tumor formation. Tumor formation. Right? So these Vir proteins, actually, their function is to recognize tDNA, which we discussed earlier, right? And in the tDNA, specifically its left border and its right border, right? And where these Vir proteins, they actually create cuts in the left border and right border and release the tDNA from the Ti plasmid, right? So this Ti plasmid here, it is linear DNA strand, and it then further transferred into the host where it causes the pathogenicity, which leads to the Crohn gall disease, right? So this natural mechanism of the Agrobacterium tumefaciens, it is exploited for the gene transfer for our purposes, right? So in the region of tDNA, gene of interest is incorporated without disturbing the Vir regions, and then this whole construct is used for the gene transfer in our desired host, right? As you see in this image, so this tDNA region is actually, it is incorporated with our gene of interest, and then same mechanism, natural mechanism, it is used for the transfer of different type of genes. So we can again review the strategy, how it works. So this Ti plasmid exists in the Agrobacterium tumefaciens. It is extracted from there, and the desired gene of interest is incorporated into it, making it recombinant Ti plasmid. And then this Ti plasmid is introduced into the plant cells, where they actually release the tDNA, which is carrying the gene of interest, and causes the propagation of that particular gene. Right? So this is how The TI plasmids are used for the gene transfer, and this is the indirect method of gene transfer in the plant specifically. So that's all for the today. Thank you.